after he finished his stint as Secretary of Energy. And in his independent lab, his interests are very much after my own heart. So he combines quantum engineering and biophysical imaging, and he's part of a recently funded a QLCI from NSF on sensing of biophysics and bioengineering. So we can't quite take it away yet. Let's see. I've streamed. We're in business, so I just need to read this thing, and then we can start. These events are being recorded and will be live on the iQuest YouTube channel. For bandwidth purposes, we would like to request you keep your microphone and video off until the end. Should you have any questions during the presentation, I will watch the chat box and interrupt the presenter at the appropriate time. Otherwise, please feel free to ask your questions following our speaker's presentation. Thank you. And that's a reminder for me to try to log in on it. Yes.
Great. Thank you so much for the invitation and for the introduction. This is actually my first in-person visit to a university since the pandemic. So 3 months after I arrived at the University of Chicago. So this is a really exciting, and it's also an exciting time because we are getting out the first results from my lab that I would like to share with you. Also get some suggestions and insights. So my lab, as Michael said, it's a very similar, is focusing on developing nanoscale probe for studying biophysical processes. Working. I'm kind of intrigued by the of biological systems and by the inherent nanoscale size of these systems. So here I'm showing an example of an electron micrograph of bacteria cells. These are actually bacteria cells that cause urinary tract infection, so they are kind of a bad thing. But if you were to focus in a 100 by 100 nanometer area, and this is our studying. Proteins in a cell. And inside this stadium, you will conclude. Should I stand in a different location? Or? I think. I guess that's the challenge, being the second person up. Okay.
I don't know what front is. Maybe front is here.
Oh.
Can you hear me? Stand here.
Oh, okay.
Sorry.
I usually prefer to stand in front of the desk. It looks like I'm defending myself this way. I guess I can choose either connect to the live or connect to the people online. So. Okay. Well, a single particle, in this case, a single person, then observe what that particle is doing, and you can learn of tennis, and indeed realize that they are very complex and interesting, actually. Measurements at single particles, at single molecules, have a huge impact on our understanding of life, on biology. So it has led to better understanding of protein conformational change, protein interaction, gene expression, motors. So, given that being able to observe individual proteins at the nanoscale, biophysicists have a lot of thoughts on developing technologies that can actually allow to do that. And made a huge impact on biophysics. Actually, in fact, each of these has gotten a Nobel Prize. In the last about 20 years or so. Actually, on using classical physics, they rely on electrodynamics or Newton. The laws of quantum mechanics to develop sensors that can do better. And that's something have already shown over and over again in the past. Showing a classical oscillator that was used until about the mid of the last century to build very accurate clocks at the time. Aid in relative precision. But once people realized that they spectroscopy on these atoms using sensitivity to field it allows us to develop a GPS system, synchronization in telecommunication, and many more exciting applications. So what my group is interested in is asking the questions whether we can use technologies that are based on quantum systems, quantum metrology, to perform precise measurements and use those to probe the nature of biological target systems. So my group indeed has kind of two main efforts. One is pushing the limits of nanoscale sensing, and then take those to biophysical targets. Basically, you take a quantum sensor that, similar to an atomic clock, allows you to perform a very precise measurement. But this quantum sensor is inherent nanoscale. So you can, in principle, bring it very close to a target system, for example, an individual molecule or a cell organelle. And then you can detect type sensors on the sensor caused by local magnetic fields or electric field or temperature. So our lab, we are not using in an ultra-high vacuum, what you use in atomic clocks, but we rather use crystallographic defects in diamond that in some sense behave, that we can put in a confocal microscope at room temperature, in some sense behave very similar to individual atoms. So these defects that we have color centers. They are in the ground state electron spin triplet that we can, under optical excitation, read out the spin state. Even if we start in a thermal mixture, to initialize to MS equals 0 projection. Atoms, we can get spin initialization and readouts through optical technique. Each of these dots is an individual NV center. And we can probe coherence in these systems and perform spectroscopy. For example, in the -1 projection. And similar to atomic clocks, we can perform Ramsey spectroscopy, and from this perform very precise measurement on this transition frequency. The transition frequency is depending on magnetic field, and so by performing an accurate measurement here, we can deduce something about the local magnetic field. So today, the sense order at best for a single NV center on the order of about 10 nanotesla per square root hertz. So these ideas are now already quite old. They're about 14, 15 years old. They were developed all around the same time at Stuttgart, Harvard, UCSB, and IBM. So what I would like to do in today's is give you a flavor of the type of approach that we are using to interface biological target systems, and how we can further improve sensitivity correlations in quantum systems using engineered highly correlated states, about potential biomedical applications that we are currently pursuing. Quantum system with a biological target, and how that could eventually I can't take off my mask, I'm sorry.
It also might turn that mic off, because I think it might be hearing the echo of that mic, and I think everyone in the room can hear it.
This improved. So the idea behind these experiments are that we can use individual centers as nanoscale magnetic field sensors. So they are, as I mentioned in the beginning, very sensitive magnetic field sensors. And by a molecule that has different nuclear spins, these nuclear spins produce a stray magnetic field. And if we use the right type of spectroscopy on this NV center, on this quantum bit, we can actually learn something about the transition frequencies of these nuclear spins, and in principle learn this way something about the structure of the molecule that we are looking at. Bulk NMR ensemble measurement that people as a standard do in physical chemistry. So these ideas have been around for a while. There had in fact been a tremendous push towards taking this classical NMR spectrometer that requires something like 10 to the 15 nuclear spins, and kind of miniaturize it to the level where you can detect individual spins, or maybe or at least small ensembles of spins. Integrate this into nanoscale sensing device. The applications of such a technology that has kind of motivated people range from single cell screening for metabolomics to studying processes in heterogeneous systems using NMR spectroscopy, and I'm sure the most interesting technology is even not discovered yet. So over the last couple of years, there has been a lot of progress towards this. So on one hand, this is an experiment that I show from Victor Acosta's lab. What people are able to do now is perform NMR spectroscopy on small ensembles, on picoliters of volume, with spectral resolution that is good enough to actually assign structures. And on the other end, there is this work that came out from Michel Lukin's lab a few years ago now, where people showed that they had good enough sensitivity to detect the presence of an individual protein, in this case, ubiquitin. The spectral resolution here is not good enough to learn something about structure, but nevertheless, it showed that they can detect that inside of a protein, you have a single protein, you have carbon-13 labels and deuterium labels. So that's kind of exciting, and it inspired the imagination of people. But what you really want to do is you want to attach an intact protein to the surface of a diamond and learn something about the complex structure of a protein and its behavior, its mechanisms, its dynamics. However, current experiment, they actually just show that you have carbon and hydrogen in a protein. We already kind of knew that, right? It's like saying we want to understand the engineering of a car, and we can tell that there is iron and plastic in a car. It's good because it shows we have the sensitivity to do this measurement, but there's still a long way to go from here to here. The other thing is the way so far people have immobilized biomolecules on diamond quantum sensors is through a way that actually completely messed up the structures of the protein. So it's really, currently we are at this stage of studying a car rather than looking at the actual dynamics. And so this has kind of motivated my lab to use technologies and tools that were developed in single molecule biophysics over the last 20 years and kind of transition these technologies to our quantum sensors. And so the challenge here was why people have been not able to connect diamond-based quantum sensor with biomolecules is the following, actually. It's a chemistry problem. On one hand, hydrogen-terminated diamond has been known for actually about 20 years, how we can modify the surface chemical bonds and attach actually intact biomolecules. On the other hand, the problem with hydrogen-terminated diamond is that the spin coherence is actually really bad, preventing any quantum measurements. I'm going to come to you in a second. On the other hand, the best quantum sensors, they rely on having oxygen-terminated diamond, this kind of well-regular ether groups on the surface. They can have very good spin coherence for the quantum sensor, but they tend to be very difficult to chemically modify, and in fact, nobody knows how to do that at the moment. So that's where the state of the field was when we started to work on this. Yeah, there is a question back there.
So on previous slide, the center.
Yeah.
And then also you said you do NMR, you said it.
It's more because I wanted to make up for time that I lost earlier. So in these experiments, what we actually do is our NV center is just a sensor. Magnetic field. So basically, there are a lot of technical details. Because replacing this quantum sensor is not that straightforward. But we can talk about this later. Okay, good. Can you hear? Okay, let me just continue. Yeah.
Hydrogen is nuclear spin and oxygen is not.
No, the thing is actually the hydrogen pins the chemical potential in such a way that the NV center that loses its negative charge.
Oh.
And so it doesn't have the optical and spin properties anymore that we need for our sensor.
So it's not just no.
Yes, it's not NV minus. Yeah. Was the state of the field. And so we thought, let's not try to mess up with the hydrogen surface. Let's not even try to modify chemically the oxygen bond, because my group is not the chemistry group. And people have tried that. The results were not impressive. So what we decided to do instead is we decided, let's keep this well oxygen passivated diamond surface, and let's add a very thin layer of aluminum oxide deposited with ALD. Just about 2 nanometers thick ALD layer does the job. And then this ALD layer has a surface that we very well know how to chemically modify and how to functionalize. So we did a siliconization and then passivated with PEG molecules. And by adding different PEG groups, we can actually control the density of functional groups. And this way we can control the absorption of target biomolecules on our surface. So here on the side, you see just fluorescent single molecule microscopy image where we use fluorescent label of proteins, and we image the density of these biomolecules on the surface of diamonds.
How deep are the NVs that you're using?
The NV centers here, they were somewhere between 2 to 7 to 10 nanometers. We add, by adding this kind of functional layer, we add something on the order of 5 nanometers. But it's really worth it because we passivate our diamond and we can actually control which molecules we want to fish out of solution. And furthermore, we were able to show that the biomolecules that we've attached, they maintain the biological functionality. Yeah. Yes, it does impact the coherence, and that's actually, I believe, on my next slide. Yes. So this technique is only good for coherence of the NV center. Does not get impacted too much. Otherwise, we could just stay with the hydrogen terminated diamond. So what we've done is we looked at the same NV center. And we could get something on the order of a few tens to, in some cases, up to 100 microsecond coherence for NV centers before and after the functionalization. There is a little bit, the red data is after functionalization, there is a little bit of a hit. And actually analyze this as a function of depth before and after functionalization, and we get about a factor of 2 average. Sometimes a little bit more and sometimes a little bit less. Coherence time in sensitivity by about a factor of square root of 2, about 1.5. So we can image this in terms of, or we can modify this plot in terms of sensitivity as nanotesla per square root hertz. And we can even discuss how long we would need to average to detect a single hydrogen nuclear spin with a signal to noise of 1. And so we get pretty close to about 1,000 second integration time. exactly above our sensor, so no lateral displacement. And improve these numbers quite a bit further. Something people have already shown in a different context. So that these are actually more pessimistic estimations in that sense.
Do you have a...
That's a very good question. That's actually what we are currently working on. So what we've seen is it's not the nuclear spins in the aluminum oxide, or the hydrogen nuclear spin that you have on the surface always have actually more nuclear spins than the aluminum oxide. And we can actually run quantum sensing NMR spectroscopy to show that the contribution from the aluminum nuclear spin is less than from the hydrogen. However, what we did observe is that the aluminum has a certain density of paramagnetic defect. It seems to have electron spins in it, and we can see those in double resonance experiments. And interesting is electron nuclear spin. And so while it looks here that this decrease, it's not terrible, but while it's a bad thing, you can now actually start using this electron on the surface as a reporter spin. And interestingly, the control over these reporter spins, we believe, in the aluminum oxide layer is much, much better than other reporter spins people have been using with NV centers before. So we are actually quite excited. We are actively pursuing right now. Okay, so we've shown that we can control the binding of our quantum sensor. We've shown that after doing all this chemical processing, our quantum sensor still has good coherence properties. Now, the next step, what we wanted to do, is show that actually the biomolecules that we attach, they still maintain the biological properties. What we've used for reasons in a couple of slides is we used short snippets of DNA called aptamers. These aptamers work very similar to an antibody. They can grab, they can tightly bind to proteins and grab them out of solution. What we've done is we've immobilized proteins this way, and through single molecule fluorescent imaging, we can indeed verify that each of these spots is an individual protein immobilized. So this kind of step function is you image with an EMCCD camera in a wide field microscope, and you see fluorescence from your protein, kind of constant, and at some point the fluorophore on the protein undergoes chemical change and it loses its fluorescent properties, and that's what causes this step function. So this is kind of the hallmark that people in single molecule biophysics have been using to confirm that they indeed look at individual molecules. So we can even do statistics now that we have this kind of binary counting how many doubles we have and how many triples. It's kind of what we would have expected. And then we can even confirm that this process functionalization remains stable under physiological relevant conditions. Meaning now that we can kind of count how many molecules we have, we can leave our sample at room temperature in buffer solution and just observe how fast these spots disappear. And what we see is that 1/d decay time is about a week. Honestly, the functionalization at this point lives longer than what most biological samples would. Yeah. However, we haven't correlated. We do see that these molecules here, they stay over the course of a minute at the same location. We haven't gone to the extent of this, but they are covalently bound. At some point, the layer starts to remove, and that's why we see that some of them disappear over now we are actually working together with a company called SomaLogics to fish out of solution specific target proteins. And so they are a diagnostics company, and we are working together with them to kind of, you can really hardly see it on this slide, but we are working together with them to immobilize different proteins on our diamond surface on demand. And so one particular application, part of my talk, but one specific application is the following. So if we can pull out of solution, what can we do with this? And so one of the reasons we are excited about this is because we can measure out the full proteome. So the proteome is measuring the entire concentration of all different proteins. That is relevant, in some sense, provides more information than genomics. So if you measure the genome of a person, it really doesn't change over time, unless if you have a cancer or something like that. So for example, young Schrödinger has exactly the same genome as accomplished Schrödinger, but their proteome is completely different. So if we can detect the proteome, we can maybe actually say something about the current state of health, whether you're close to a cardiovascular arrest, something like that. At the same time, most pharmaceuticals, they act actually on the proteome, not on the genome. So the hope is if we can have a high throughput technology to map out the proteome, we could use this for diagnostics and for the development of new pharmaceuticals. However, the problem is, while there are really good genomics techniques, there isn't a good proteomics. And so we are working together with this company, SomaLogic. It has developed a biochemical assay to do high throughput proteomics. The problem with this technology is it's very demanding. It's a general, it's kind of a traditional molecular biological technique. And it takes a full-blown molecular biology lab. So what we are hoping is with quantum sensing to kind of cut down all these washing steps, make a chip-based technique that could be significantly improved. And the idea here is the following, that we have different measurement sites with our quantum sensor that locally measure binding events, so snippets of DNAs that have paramagnetic spin labels in it, and these paramagnetic spin labels undergo a chemical shift depending on binding events, and this way we hope to be able to locally distinguish specific from unspecific binding. So this is actually something that we are currently pursuing together with this company SomaLogics and with Stefan Stoll's lab at the University of Washington. They are experts actually on EPR spectroscopy of biological molecules. The idea here would be to kind of integrate the diamond functionalization technique that I was showing before with this individual quantum sensing measurement station, and then eventually integrate that into chip-based technology. So this kind of concludes the first part of my talk, the quantum, how to interface biological systems with quantum sensors, which is a non-trivial task, but using tools from biophysics and quantum engineering and material engineering, we can actually achieve that. And I hope that I was able to give you a direction excites us. Now what I would like to do for the last 5 to 10 minutes is switch gears a little bit and talk about a completely different project that started because of COVID, because our labs were shut down. And the question that we started to ask is, how far can we push nanoscale quantum sensors? So quantum sensors that consist of a couple of spins, maybe up to 10 spins, can we use other sensors? And so the idea is here the following. Again, if you have an NV-based sensor and you have a biomolecule on top of it, you can put a spin in between, to kind of a reporter spin in between to cut the distance off for the molecule that you want to send. People have been theoretically proposing this 10 years ago and done experiments to that. But they actually do better than this. In principle, yes, your sensitivity could go up with the square root of the number of spins that you have. That's known as the standard quantum limit. The question is, can you do better than the standard quantum limit, better than the square root of n scaling? The answer again is yes, in principle. You can use, can go to linear scaling, known as the Heisenberg limit. But the problem is actually with spins that the spins stop interacting with each other via dipolar interaction. The single particle coherence time will go down inevitably. And it turns out that the sensitivity gain that you gain from a detection volume, which is inherent, gives you the same amount of reduction in coherence. So your sensitivity will kind of flatten out. So the question that we were asking, of highly entangled state, may bring us to recover Heisenberg limited sensitivity. And so our approach here was the following. We have an ensemble of spins. They didn't talk about direction. And we have quite limited control over the spins. So we don't have single qubit addressability. All we have is single qubit readout and kind of coherent manipulations on all spins at the same time. We can apply pi half pulses or any rotation on all the spins, but all the spins at the same time. Certain duration on the dipolar interaction, here indicated with a D. And so what we do is we make an ansatz of a certain set of gates, and we can talk about what the motivation is of this, but let's take this for granted just now. And we then can optimize parameters in this set of gates based on the outcome of our measurement, our single qubit measurement. And so kind of optimize this circuit through a feedback loop. So that's kind of a variational algorithm as an answer. And so what we can find is that actually what is complete spin system, that the sensitivity goes up well beyond the standard quantum limit as we increase the spin number. There is kind of a flattening off, and we have a pictorial or a phenomenological understanding why that happens. But as we increase the number of repetitions on this gate set, we can actually push this higher and higher closer to the Heisenberg limit. So we can actually study the scalability of this dipolar interacting spin, and we don't have exponential and polynomial scaling, but for small numbers of spins, we can actually, with resources of up to a circuit depth of 7, we can actually get within a certain percentage of the Heisenberg limit. So we can even repeat this. There we can actually get even closer to the Heisenberg limit, and we think the reason is that we actually can take advantage of symmetries in our spin network. Yeah, exactly. Yeah. It's a limited number of spins. It's all 10 spins here that are in our nanoscale sensor that we can pack into our nanoscale sensor. And those spins, they don't redistribute. They are in a position, and we can kind of, if they redistribute, that would be a problem.
So if you're
So yeah, let me see if I have actually a slide for that. I have in a slide also. I have I discussed this. It's kind of here, we already have to deal with the anisotropy of the dipole interaction. Depending on your spin configuration, some spins can have actually almost zero coupling. Compensate for that is by going to a deeper and deeper circuit, so that you do operations that allow over this. So get even a little bit better feeling of what activity. And for the square array, where we get very close to the Heisenberg limit, this is not surprising. We get out the GHZ state. So this is the Wigner distribution for 7 layers. And for the random configuration, we go up 6 spins, where we are still close to the Heisenberg. Have also a GHZ state, but then as we increase the spin number, this starts to turn out to be closer to a spin squeezed state. And yeah, this is addressing your question. If you take just 2 layers, what I've done here is each of these points is an individual spin. The color indicates the amount of von Neumann entropy that spin has with the remaining set of spins. And for 2 layers, some of the spins have quite a bit of entanglement, but many of the other spins remain almost unentangled. And so we can, this is only a single particle entangled with some part of the ensemble. We can characterize a little bit better by measuring the von Neumann entropy for the smallest set, and what we find is that for small layer numbers, this kind of ensemble of spins kind of disjoins in multiple clusters. Versus if we go to 7 layers, most of the cases we have just all the spins kind of entangled with each other. We can then also study this on the performance of noise, and what really is, I would say, experimentally probably the most challenging part to implement this in a solid state system is readout fidelity. It's not very surprising, but your readout fidelity is really what can kill you. If you have a GHZ state, if you have one single readout error that is a mistaken parity measurement, which kind of messes up your entire optimization algorithm. Oh, I'm missing this slide. So we actually can also optimize our algorithm under the presence of readout noise. And what turns out is if you have readout noise, at some point you start not to optimize anymore on a GHZ state, but you optimize onto a squeezed state. Somehow that slide got lost here. And
is that like a squeeze state, or there are lots of other states in between?
Exactly. So there is this middle ground in between. And a large spin number. You go. There's also this area in between, either in a squeezed state number. Still have actually better sensitivity than just a squeezed state. A little bit unfortunate that light fell out, but yeah, if you optimize your algorithm, you actually, depending on a realistic noise model, you can end up with just a squeezed state optimized if your readout errors are finite. At the same time, actually, your coherence times the nearest neighbor dipolar interaction strength. If you're in the regime of about 10, the coherence time is 10 times longer than the inverse of the dipolar interaction strength. Then we are definitely above the standard quantum limit. So the dash down here is the standard quantum. With promising systems could be defects in diamond, where people already showed like 95% readout fidelities and long coherence times. And actually, I think also cold molecules could be an interesting. Yeah, Paul?
Is
Yeah. So in the let me go back here actually. For a ring, it oscillation. And I think it has something to do with that you can never have a case where all the spins are kind of optimized with each other. But I think in this I think it's actually more a finite size effect because our optimization with noise were kind of computationally expensive. And so I think this is just deadlock in this case. The other thing to add actually is there are these famous theorems that entanglement doesn't help you in terms of sensitivity if you have a Markovian noise environment, which is actually an interesting thing for or solid state systems. There you often have a non-Markovian noise environment, and in that case you can actually get an improvement. So that's another thing I think that makes this technique kind of interesting for diamond sensor or solid state sensors. So yeah, with that I'd like to close with kind of the skyline of the city of Chicago, and I'm really lucky to be at the Pritzker School of Molecular Engineering because it combines kind of the quantum engineering aspect with immunoengineering and materials, and that's really what kind of drives our quantum sensing research. I'd also like to just talk briefly about our new NSF Quantum Challenge Institute, Qube, that is focusing on developing new quantum sensors for biological sensing applications. And so our approach here is to develop new Qubit sensors, either diamond-based or the next generation. And combine it with new type of imaging technique, and then work together with groups in the life sciences and in the medical school to actually use it for biophysical and medical devices. And again, what our colleagues in the life sciences say can actually inform us back on the sensor design itself. So with that, I'd like to end with a picture of my group and where my lab is. And I do have actually an open postdoc position for somebody who would be interested in developing an NV-based nanoscale sensor at actually large magnetic fields of 14 Tesla. That would need terahertz